think this is probably everybody we're going to get for now, unless we have latecomers.
Yeah, I agree. Michal, there you go. Would you like to start?
So yeah, Michal, I suggest we start, and then you know, since they're less than we expected, we can end earlier.
That's perfect with me.
Okay, so
You want to introduce yourself, and then I'll introduce myself, and then we'll continue with the short presentation.
yeah, so perhaps people who can present their name, where they're coming from, and what's their main interest in neurodegeneration, neurobiology, so we can better understand exactly how to organize the discussion and who to point different questions.
We'll start from that side, I guess.
Okay. So I should start with the introduction. So hello, my name is Schwartz. I come from a company called Bit Biodiscovery, and we are using forward programming of cells into neuronal cell types, and we also model diseases and then use the healthy versus the diseased cells to perform CRISPR screens with a single cell RNA-seq readout.
Nice to meet you.
A little bit closer. It'd be a lot easier for you to pass this around yourselves without the
Yeah, I'm Jürgen Knoblich. I'm here from Vienna, from the Institute of Molecular Biotechnology, and I work on neurological disorders, particularly on their effect on brain development using in vitro disease modeling.
Özgün Gökçe, Munich. I work on the single cell and spatial transcriptomics methods to study neurodegeneration and aging, focusing mostly on the white matter loss and the myelin degradation during diseases.
Jason Hilton, I'm at Stanford and I lead a data coordination project. And so we do data wrangling for the HCA seed networks, pediatric networks, and ancestry networks. And we also lead curation of data in the Cell by Gene portal.
Hi, I'm Katie Geiger-Schuler. I'm a senior scientist at Genentech. I'm really interested in both profiling and high information content multimodal genetic screens to study mechanisms of neurodegeneration.
Wei King Tay from EMBO EBI. I work with the Human Cell Atlas data coordination platform, trying to get high quality metadata from all the publications we can really get, and just here to tune in and listen.
Thank you. I'll also briefly introduce myself. So I'm Rob de Horst from SEM here in Vienna. I'm a bioinformatician postdoc working on different types of data, both bulk and single cell, and I'm interested to hear what everybody has to say.
Hi, I'm Yi Kwan. I'm also here at SEM. I'm part of Julio Superthorger's lab, and I'm interested in the interface between proteostasis and metabolism in neuroscience.
Ido, do you see my screen?
can't see your screen at the moment, Michal. It goes into
Can you see my screen?
No, not yet.
Michal.
So there is a problem all of a sudden in my.
Michal, maybe there is a recording message. Just click approve, and then I think you will be able to share.
No, I think that because there is an electricity problem here, I need to reconnect.
Okay.
They're fixing the electricity in our building right now, so I'm reconnecting.
Okay, you can also send me the.
No, I'm fine. I just need to reconnect.
Okay, we will. I'll introduce myself while you're reconnecting.
Okay.
All right. So, it's great to have everyone. Definitely the neurodegeneration field and understanding in general the wiring and various cell types that encounter brain normal physiology and especially during many diseases which are unresolved is increasingly becoming more and more important. The HCA and the many contributors to the HCA, I think, can make a very large impact. There are many challenges. So the way we're going to try to have this discussion is in a way that first Michal will introduce some major history points in this scientific area. Short introduction of around 10 minutes. And then we're going to have an open discussion where we really want everyone to share and make this very important bonding that makes the HCA so special, to talk about their project, to talk about their challenges. There are some common points that I think are very important that we would love to hear people's input. Some of them have to do with the way we are currently annotating cells in the brain. There is very large projects, both from the NIH, EU, and others. And I think one of the major challenges that everyone is struggling, both in general, but I think also very specifically in the neurodegeneration or brain mapping, is how do we actually annotate the cells? How do we make a communication where everyone talks about the same subsets of cells, same markers, and so on and so forth? So it would be great following Michal's
I'm still struggling with it, so keep on for a few minutes and I hope to solve it. My computer is completely stuck at the moment.
No, it's great. So you know what, Michal, we can, if you want us to really start having a discussion on this.
No, I can start without the presentation. Also, I prepared presentation. Hopefully, if you can hear me.
We can hear you very well. So let me just finish the point. We can hear you well. So let me just finish the point and then let's go to your intro. So after Michal's intro, I think it would be wonderful to have group thinking and again, potentially making important links between people working on this area of where do we need to improve, how do we make better connection, how do we better annotate and understand the various subsets of cells in the brain, understand their biomarkers, understand how their biomarkers and cell states are changing throughout disease. And I think that would be one good area if we can make progress in today's breakout session. So without any ado, Michal, the stage is yours to put everyone on the table on the really burning aspects in neurodegeneration and how single cell can really make a significant ground on this aspect.
I think she might have just fallen off.
Maybe it's a good thing because I think when she connects, you will likely be able to present. So I think that may be. So why don't we, until Michal joins, I'm sure she will find a solution. Why don't we discuss, are there anyone here generating or analyzing large data sets regarding the brain or the brain under different pathological conditions? Yes.
I do, and I'm Özgün Gökçe, and I do. You actually gave me the best advice when I start my group in Belgium. You said, just say no to everything. And that actually helped me achieve a lot. Thank you.
Okay. You know, sometimes when people say this, I'm worried. I said it when I was drunk and I said something wrong. So
we were going to restaurant, you weren't drunk, and it was a good advice. It was really letting me focus things. And now we use spatial transcriptomic and single cell genomics, white versus gray matter. And congrats for the oligo paper. I think this the path you open with the DAM and now with oligodendrocytes. I think these are important directions. I was wondering what you think about, because now you move to do a lot with the cancer and we all became immunologists with the actually Michal and yours leading the path for us. How do you see cancer? How much things you learn from the cancer research bringing the neurodegeneration?
Yeah, well, it's a good question, and maybe we should open it up. I see Michal is back on, so I'll just answer, and then I think Michal will be on with the intro, and then later we can continue. I think, you know, once understanding different pathological immune pathways across diseases is something that doesn't, I would say, stall or delay progress, but really makes progress faster. For example, we see many common pathways which are shared between immune dysfunction in the brain and immune dysfunction in cancer. TREM2 pathway is one very obvious one. Obviously, it's very difficult to understand what would be the outcome of modifying these pathways in the two conditions. And there is a lot to make then progress on functionally understanding the different immune context, what you actually would want to modify, what is going to improve, or in some cases, God forbid, actually make the disease go worse. So we'll get back to this. I think we have Michal online before the electricity drops again. Let's have the introduction, and then we can continue the discussion. I think there's a lot of such discussion, and we'll pick up.
Michael, she's muted. You are muted. Michal,
you're muted. Can you? Michal, you're muted. Still muted, Michal.
Now.
Perfect. Come on. The presentation. Yeah, great.
You see my screen now?
Yes. Perfect.
Perfect. So we can see
your screen.
No screen. He's not sharing.
You're not sharing it, Michal.
Wow. So I'll start all over again.
She was sharing.
Okay. Yes.
Okay. At least you can see my screen now. And I'll show you the presentation in a few minutes. Okay. So can you see it now?
Yes.
Okay. So I think we don't need it. Right now, we know that there are numerous neurodegenerative diseases, and there are have several in common. First of all, and I'll show you here, that most neurodegenerative diseases are sporadic and not genetic. One good example is Alzheimer's disease. We now know that the major risk factor is aging. What is very important to be aware of, to the good and to the bad in terms of scientists, that these diseases are very dynamic. There are multiple factors, but the most important is that they are associated with misfolded protein, which are features by the presence of aberrant protein, including amyloid beta oligomer, plaques, neurofibrillary tangles. What is also important that we learn from Alzheimer, and it's applicable to many other diseases, that the symptoms arise relatively late. In Alzheimer, fortunately, we have still a large window of intervention after symptoms. In many of these diseases, such as ALS, and even worse in Creutzfeldt-Jakob disease, once the symptom arise, there is very little window for intervention. This disease are speedily progressing. In ALS, it's like 2 to 5 years. In Creutzfeldt-Jakob disease is 1 to 6 months. So there is a really urgent to understand more these diseases in order first to identify early biomarkers that could serve as target for intervention, and to understand that these diseases is not just the protein pathology, but there are many beyond that. And one factor that we learn in Alzheimer, and it's applicable to all the neurodegenerative diseases, that there is a local inflammation. And I brought this example because it was published a couple of months ago in Nature Medicine in clinic. They demonstrated that you may have amyloid plaques and no symptoms. You may have loss of neurons in the form of tauopathy and no symptoms. And only later stage, when you have both amyloid beta and tauopathy, there is loss of cognition. And it turned out that there is a huge synergy with the missing link, which is the local inflammation that they call it in the paper as activated microglia, which tell us that we are far from understanding the role of microglia. We are far from understanding how far they are contributing, what is the stage that they are beneficial, what are the stages that they are not beneficial and even destructive. So this a key factor, missing gap between what we understood so far is what will lead to finding cure for these diseases. So what we knew for many years, the disease was considered, all the neurodegenerative disease were considered as neuronal centric. Now we know that it's not neuronal centric within the brain. It includes astrocytes, oligodendrocytes, and neurons. They are not brain centric. We now know that there is a huge role for immune cells. We know that there is involvement now for peripheral immune cells and niches within the brain that were lately identified by several groups. Among the leading group now is our group in Israel, the group in St. Louis by Jonathan Kipnis and Marco Colonna, the group in Harvard, the group in Stanford. All of these groups are aware of the role of the immunological niche that we were not considering at all. And of course, we need further to characterize what is the function of these niches, the choroid plexus, the meninges, the cerebrospinal fluid, and the lymphatic vessel. And now we know also the bone marrow. So currently there is no single therapy for any of these diseases. The diseases are dormant long before symptoms are seen. The symptomatic stages differ from one disease to another. They may be short and long. So I think that the revolution in understanding the brain relationship with the immune system is what will be the future in neurodegenerative diseases. And I heard the question about what one can learn from cancer to neurodegenerative diseases a lot, because neurodegenerative diseases were considered as diseases of the brain, as I said. Tumor were considered as disease of the tumor. We now know that the immune system is a key role in fighting against against cancer, and this will be the future in neurodegenerative disease. So just to give you the flavor of the field, if we go back to the history, in 1906, Paul Ehrlich introduced the idea that the brain is compartmentalized from the periphery, and it's a closed compartment. And this led to the immune privilege, to the idea that the brain is separated from the immune system. And I think that Medawa, when he got a Nobel Prize for coining the idea of immune privilege, that was the last point that put the field into the axioma. The brain is isolated from the immune system. And it took decades to break this dogma. And I think the breaking of the dogma was summarized very nicely lately in an article in Nature, where now, rather than consider as the enemy of the brain, the immune cells are the guardian of the brain. So we are now aware that there is a bidirectional relationship between the brain and the immune system. What was considered as barriers between the circulation and the brain, the blood CSF barrier, the meningeal barrier, and the blood-brain barrier, has gone revolutionary over the last few years. So the barrier, the real true barrier remains the blood-brain barrier, but the blood CSF barrier and the meningeal barrier are now a barrier that are considered as site of interface. So the brain is still immune privileged site, but their standing is different. So what we have learned over the last few years, that microglia are the immune cells of the brain. They are perfectly great cells as long as we are healthy, but they are kept under strict control by several molecules, among which are the fractal kind, the CD200. We learned that the single cells has advanced the field very nicely by initially by our work on microglia, the disease-associated microglia, later on by the astrocytes associated with neurodegenerative diseases, and lately with the oligodendrocytes. So all these are great, but still we are missing the understanding which changes are for the benefit, which changes are not for the benefit, and which changes are even destructive. Can we control it? So I think that we have now a room for many intervention. We have a room for learning a lot from the single cells in all of these diseases. There are very recently articles that show that the immune cells in each of these compartments are beneficial and could be destructive. And I would like to point your attention that in 1998, when we first introduced the bone marrow derived macrophages are needed for the brain, it was considered as forbidden because at that time it was considered the brain is immune privileged site. Now with the recent observation, we are not only convinced that the bone marrow derived macrophages are needed for the brain, but there is a direct communication between the brain and the scalp via the CSF. The initial paper was published by Niederhartz in 2018. Recent two papers in Nature Neuroscience show that not only there is communication between the brain and the scalp bone marrow, but the CSF is flushing them. So we think that the new understanding that the privilege of the brain is not being disconnected from the immune system, but control what can be in and out in this communication. So I think that with this, we can open out a discussion to understand what is the future in this field of brain immunity. I think this is a new era in searching for new therapy for neurodegenerative diseases. I don't want to take more time. I have many more slides, but let's switch into discussion because I know that Ido needs to leave very shortly. So Ido.
Michal, really thank you for putting everyone on some of the canonical and really important areas that we need to make progress where more open questions than answers actually. And I think we were, just before you talked and you touched briefly this connection to the brain and how could other pathologies, both inflammatory, for example, in cases of MS, as well as cases of immune dysfunction, can really teach us a lot about what we should or could expect when we may move into functional question for these pathways in CNS pathologies. And of course, this is an area of huge interest by our joint groups, as well as many in the field, some you mentioned. So I think this is again open. We want to really hear where people are seeing challenges, or even more important, where do people see areas where the HCA can help by forming really stronger allies between the different efforts that are being done. And I don't see anyone from the EU effort, but there's a very large EU effort and obviously NIH efforts and others. So let's continue the discussion.
Hi, I'm Zorina Gallus. Can you hear me?
Yeah.
Yeah, we can hear well.
Oh, good. So I'm Zorina Gallus from the Human Biomolecular Atlas, the NIH say counterpart of HCA. We are not looking at the brain because there is a specific brain initiative. However, my interest is in how what happens in the brain is related to degenerative diseases in the rest of the body that are mediated through aging, not only of the immune system, but of the vasculature. And specifically for Alzheimer's, the #1 risk factor for Alzheimer is high blood pressure, which probably damages the blood-brain barrier. So I was wondering, what are your thoughts about that, and whether looking at what happens in aging in the neurovascular unit is a very important component of Alzheimer's. And
this is a very good point. This is a very good point. We have several works that we are doing, another group are doing. And the key question is indeed, now we understood that the disease is not just the diseases of the brain, but it's influenced by the rest of the body, and mainly the immune system. I can tell you that we know, for example, that one of the key comorbidity factor in Alzheimer is diabetes. And we have strong evidence from our group and others that high-fat diet accelerate Alzheimer's disease, and the major effect is via affecting the peripheral immune system. So this is one of many examples. You are absolutely right that the risk factor is high blood pressure, but again, it's not only the vascular, it's the low-grade inflammation that you have in the periphery. So I know that there are several groups that are working further characterizing how the immune system is influenced by high blood pressure, by diet, by diabetes, by obesity and other factors, and also by nutrition in general. Did I answer your question?
Yes, but I think in order to do something about this, is very important to try to figure out sort of the chicken and the egg, right? So is inflammation that starts the opening of the barrier, or is the opening of the barrier that triggers the inflammation and so forth? I think we would like to go as, you know.
Yeah, you're right, but I don't think that it's only the opening of the blood-brain barrier. I think that the general feeling and belief now that it's not the beginning of the disease. The disease is dormant for a long time, and it's not triggered by high blood pressure. It's not triggered by the diabetes. These are only catalysator of the disease. They may determine the time of onset that may be earlier, or that the disease will be more severe and progress much quicker. The common belief now that the changes in the body, or the way that you call it, or the changes in the immune system is not the trigger, it's dictate the timing of onset or the speed of progression, but it's not the primary cause. But time will tell. There are many groups that are working on it now, but the chicken and the egg, probably the disease starts in the brain.
If there are no questions, I might like to ask about the opportunity. As the atlas, what I would be interested is showing especially with the spatial genomic transcriptomics rising, a roadmap for how to map immune system brain interactions, and what would be the procedural when we are doing, what are the experiments, markers, what are the key molecules we should be characterizing, and the neighborhood analysis and linking to different cell states to neighborhood, because so many opportunities. And I feel like not only the cell atlas as a naming, but now we have the spatial map, and we can actually see which identities appear in the same space. And we can actually look to the molecules. And any ideas about how this could be implemented in the human cell atlas? It's kind of an interactome of immune brain, nervous system, immune system.
Yeah.
Yeah, go ahead.
So basically, we are speaking about several compartments. Within the brain borders, you have the meninges, you have the choroid plexus, you have the CSF. Then you have the skull, which is connected to the bone marrow. So there are key questions that we still don't understand at all. And also you have to keep in mind that the drainage of the brain is to the deep cervical lymph node. And that's where the cells are being educated. So the entire pathway, even in healthy situation, is far from being understood. We all are speculating and we are working on it, some by ourselves, some in collaboration with others, but we all know that we think that the drainage is to the deep cervical lymph node. We believe that the cells are being educated, the deep cervical lymph node. We now believe that the drainage of the antigen is via the lymphatic vessels and the CSF, or the entire CSF. So we think that this is the primary education of the cells with respect to T cell. With respect to B cell, there are some speculation now that the education is being in the meninges. The source of the bone marrow, of the B cell, is probably in the bone marrow. So because before we are going to special, we need to further characterize in each compartment, which of the cells, where is the education, the site of interface, where it's going to be, what happened in disease condition. Is it the drainage? Is this that within the deep cervical lymph node there are suppression cells? Are the checkpoint inhibitor going up? So there is, the human cell atlas will help us a lot. Once we identify the cells, we can compare to the knowledge in order to know which are the cells. So I think Ido can speak about sharing of resources, sharing of knowledge, but the brain immune interfaces, we are just starting to understand. Some of the work emerged from the Weizmann, some from other laboratories, but it's really, we are really at the beginning of understanding these interfaces, and they are not uniform. Probably each of them have different job, and how they communicate is also not clear. Because if the brain is connected to the skull via the CSF, and the CSF is flushing the bone marrow, do we see the macrophages that we see in the perivascular spaces? Are the macrophages coming from the skull bone marrow? Are they coming from the periphery? Do the skull bone marrow differ from the bone marrow in other places? Following spinal cord injury, we see that the macrophages can enter through the leptomeninges adjacent to the site of injury, but they're coming also from this choroid plexus. So there is a lot which is unknown, and the human cell atlas, Ido can speak about it, how it can help us to move the field forward.
So I think I just want to add, and actually I would love to hear people from the crowd more than just Michal and I answer a question. So it should be like an open discussion. But I just would like to add, I mean, very specifically on your question, that one of I think the challenging in how we should think about designing the CNS and especially CNS pathologies within the HCA is we shouldn't think about only the biopsies from the brain. We have to have connection between samples that we're taking from the brain and samples that we're taking from immune periphery. Okay, and I'm sorry to be immune focused. I'm sure there's other aspects that we should think about, and we currently don't have this in our I think in our thinking. Our thinking has not introduced the physiology, which is, as Michal, beautifully introduced and dreamed about this so many years ago. It's all connected, and if we're just looking at the brain, and we can never make these association to the pathologies that are actually maybe initiated, maybe happening in a consequence, maybe as feedbacks. If we don't incorporate this, we're losing very valuable information, and we should think how to really make measurement that couple the brain pathology with the periphery. And I think this is really maybe the next frontier where we should really think about going.
I would like to add, though you're absolutely correct, that it's more critical even the CNS that in cancer, because in cancer you can do biopsy of the sick patient. In the brain, you don't do biopsy. So you have to extrapolate from post-mortem to what you see in the immune system of still live individual, and in order to connect this.
Correct. So mostly, I mean, many times we only have a post-mortem.
Yeah.
Sometimes,
and peripheral blood that we can get anytime from all these sick patients. And we need to bridge the gap, and by in silicon analysis and by many other things.
yeah, I mean, maybe you can speak
Just
about
before we has her hand up from the virtual audience, can we bring her on? Dory?
Hi, yeah, thanks. Can you hear me?
Yeah.
Yes, Zoe, please go on.
Hi, yes, thanks. I'm Dory Hockman from the University of Cape Town in South Africa. Thank you for letting me join in this from far. I just wanted to bring up the point that you just raised about being able to biopsy the brain and get information from the CSF or from the periphery at the same time. We're in a unique situation here in Cape Town, and I'm guessing there are other places around the world where we can get access to the brain tissue, antemortem brain tissue from epileptic resections.
Yeah.
And often there's actually, I guess, it's possible that there'd be CSF collected from the same patients if, say, they, or also being treated for, yeah. It's possible that we may have both of those samples. So I think that's a very interesting point you raise, and it's maybe good to get people that are in this sort of situation where we are here, where we have close connections to neurosurgeons and are collecting brain biopsies, and then considering whether, you've just raised to my attention that I should consider whether we are collecting CSF from these same patients and whether we would be able to, say, do single cell analysis on both the brain biopsies and tissue, cells extracted from the CSF.
Ido, you are muted.
Yeah, sorry, thank you. Dorit, first of all, thank you for joining from afar. You know, your point is really good. There are many like you who are making a very significant effort and have specimens which are very unique. And, you know, consider that when the HCA started, we didn't know we can get to this massive amount of measurement. Spatial was really not there. And so I think, you know, not long from now, we are going to, you know, scale up the technologies 2 orders of magnitude. Sequencing costs will drop, and efforts to now go deep into the peripheral tissues will commence. And so if you maintain, you know, it's okay today to focus on the most meaningful samples you have, and definitely the biopsies are the most meaningful samples, and we still need some, I would say, improved technologies and better understanding of the periphery to know what we are going to sample from there, or maybe we'll have the ability to do shotgun by that time. But I think that preserving them, and these samples we know how to preserve very well, can really make a very significant study today, but also that you are going to be able to adopt and improve all the time in the future. And I think that's really important, that, you know, we maintain all of the potential sample that can be taken from the patient in order to make this connection. Some we can do today, but some we will be able to do much better in a year from now, and 2 years from now, and 3 years from now. So, you know, that's a great point. Thank you for raising this.
Yeah.
Thanks. Can I also ask, in terms of like the relevance of these sorts of samples to disease pathology, because I guess that they're coming from patients who have epilepsy, obviously. But I was just wondering, in terms of later on, if you want to understand what's going on in Alzheimer's, say, if we have these sorts of samples from a different disease pathology, or maybe even slightly, whether those are going to be useful as a control for say, comparing to what's going on later in life during Alzheimer's.
Definitely, yes, because first of all, if I understood correctly, there is a high incidence of epilepsy around a patient with autism. So it can tell us a lot about the pathology of autism. It can tell us a lot about the relationship between the immune system inside the brain and site of pathology. So I don't know whether it will be useful as control for Alzheimer, but it will be very informative, whatever we will consider it as an example of control for Alzheimer or control for other dysfunction of the brain.
Yeah, cool. That's great. So I think that's the main driving force behind my group's work is to use these very precious samples, which are as normal brain tissue samples as we can get that aren't post-mortem to serve as some sort of comparison for other disease pathologies in the future.
Perfect. Any other questions?
Hi, it's Katie. I had an unrelated question, if it's okay to switch topics.
Yeah, absolutely. Katie, can you mention where you're from and then the interest? So we want to, know each other.
Yeah, so I'm in Genentech and I'm in Arit's cellular and tissue genomic department, but I have collaborations with the neuroscience department. So I'm focused on kind of developing new technologies that can help us answer questions in neuroscience. And as I talk to my collaborators in the neuroscience department, I realize it's going to be so helpful to have an atlas, but once we have it, we'll definitely have it. What do we do next? And there's issues with sort of mouse models in that like all of the microglial genes are quite different in mouse and human. And while we know that, the interactions are complex in disease, they're going to involve interactions with the immune system or immune cells and maybe neurons. If we don't have a mouse model that recapitulates the human biology, how do we go in and test hypotheses? Or how do we go in and do screens? Should we be thinking also about models to generate that are more complex or mouse systems that are humanized?
So I will start and maybe though you'll continue. I think that you're absolutely right. And one way to overcome the gap between the mouse and human is to test everything in multiple models in mice. And this is key issue because I think that I can speak from the Alzheimer field. In the past, we thought that Alzheimer is a disease of amyloidosis and it's not disease of neurofibrillary tangles. Now we know that it's also disease of neurofibrillary tangles, so it's a disease of tau, which is shared by all kind of dementia. So I think to avoid the problem of the gap between mouse and human is to check everything, not only in mouse model of amyloidosis, but also of tauopathy in order to see whether there is unique or common pathway. And I believe that when we go to the immune system, if you check everything across several models, you will see the commonality much easier. And we have it from our own experience that by checking multiple models of dementia, you see the commonality.
Yeah, so I think, there's other aspect that we should consider. Models are just models, and obviously they cannot represent the full-blown disease. Some aspects truthfully maintain some molecular aspects. For example, the dams that we see and the dolls that we recently published are quite conserved from mice models to human. Some aspects are not. So I think a full understanding of what's conserved and what's not conserved is important. I think once we understand molecular trajectories which are conserved, that allows you to think about or test different drugs at different stages of development and understand that whether they fulfill at least this aspect of modulating the specific molecular trajectory that you're interested in modifying. I think we should also think about it a bit more, I would say in a bit more modern way, in a bit more like the HCA way, which is let's think about these models. Do they recapitulate the molecular phenotype? And if not, then we definitely need to improve the models until we get one that do recapitulate the molecular phenotype. Regarding, the full-blown pathology, I think this is asking way too much from our animal models. They are never going to recapitulate the major pathological aspects, which are in humans developing in a very different way.
But maybe the commonality is the way that will lead eventually to the therapy because of the heterogeneity also among humans.
I think also as a drug company, you don't want to find something that's really great in mouse and it's not expressed in humans. So there's, we do start to care about like specific genes, and that's my concern sometimes.
I absolutely agree with you, and there are examples of that.
Maybe it's really nice comment. This is something was bothering me as well, because Michal and Idos, the peripheral immune nodes of the human brain is critically important, but they are the hardest to analyze because we get the postmortem. We don't know how long the hypoxia, and there are confining infections. And when we get this tissue, we are looking into, even in the healthy case, a really alarmed and activated state. So I think in this case, animal models are more helpful because this co-finding because everybody died and immune system responds to that maximally. So what we as a human cell atlas going to map, going to be not the normal healthy one, because when anybody died is not healthy.
Yeah, but I absolutely agree with you. And I think that we have to go back and forth from human to mice and mice to human. And it's unavoidable. I absolutely agree with you. And I think that what is nice, that also what we know now about the human disease came a lot from the mice and not the other way around. And Alzheimer is a good example that after insisting that the disease is amyloidosis, there was a recent paper that showed that even in model of amyloidosis, the 5xFAD, they observe hyperphosphorylation of tau without having any gene that relates to hyperphosphorylation, which is so nice. So the mouse recapitulate the human, and the human the mice when you look for it. For decades, no one looked for hyperphosphorylation of tau in amyloidosis. And when you search for it, you find it even in mice, not only in human.
Additional question from the audience?
There's one.
So I'm not in the field per se. I'm a bioinformatician, and as just thinking of treatment, I'm wondering. You mentioned before that often once you detect the disease, it's already quite late. Does that mean that the symptoms are not severe enough before we detect it? Are there symptoms that were not detected? Like thinking about detecting patients, when do we have a risk population that we can sample? Would activity trackers on them already give an indication that something is wrong? And then, these are the people we should do a biopsy on, because you cannot sample people, even their blood, every month, right? So what would you think would be a strategy in the future treatment of disease to, like, would people in an old folks home need to do like reaction speed tests every month? And then, these are the people we should sample, or what is your thought on that?
So I had a slide about it, but I skipped it, but I can give you a good example. This is a very good point. And mainly to diseases like ALS and Creutzfeldt-Jakob disease, that the window, once the symptoms appear, is very, very short. It's a very slippery slope. So we have data, we are wrapping it up now, that shows that if you have access to carrier of mutation, you can learn about changes in the blood profile if they happen and when they happen prior to symptoms. This could serve as a great biomarkers and potentially target if you find it. So this is one way to go. And if you do it with robust searching for unbiased markers, you can come up with other biomarkers. And this is one of the things that we are proposing for Human Cell Atlas, to go after blood samples in carriers prior to symptoms. And extrapolate, of course, to non-carriers.
Thanks.
Other questions?
Dorit, you have a question?
Yeah, hi. Thanks. I thought I'd bring something up now that we know that we have a bioinformatician in the room. One of the things that is a big challenge, I think, is for us to do the differential expression analysis that ultimately we need to do to find what's different between healthy and disease states and different stages of disease. And I was just wondering where the people in the room are at, and they're thinking of the best way to do that, because I know at the moment there's a lot of the tools are taking pseudo bulking approaches, and whether that's really the best way. Are we just losing the power that we gain from single cells, and whether anyone has any thoughts? I'm quite new to tackling this problem, so I was wondering what sort of approaches people are taking for differential expression in single cell data at the moment.
So I can start, but I think it would be great that many contribute to this. And so first of all, it's great that you jumped in and you're not afraid to go before you have everything parsed out and understand that analysis. Sometimes we collect data for half a year and analyze for 3 years. So, analysis takes time, and that's something that usually when people go into such areas, they don't consider how actually some of the analytical challenges that are going in. So definitely there are ways to move quick and dirty, to understand, where you are and how to best continue data collection. But when you really want to go deep into the analysis, then there's definitely a need, and there are definitely very good approaches to model the data and allowing you to go into single cell resolution in terms of the analysis. So, it definitely you gain some things by making these pseudobulks of presumably similar cells, but you also can lose a lot. And this is something that you really need to, continue the analysis. It never ends in reality. And Michal and I, just had a discussion about, the old data where we found that there were some really hidden gems there that we missed, and we should have gone back and looked again and again, and we would find it. It's just that, the first glimpse, you see the mountains, then you see the valleys, but there are really very nice things also in these very small fountains that are there. So that's my short. Long answer is more complex, but yeah, please. It's meant for a discussion.
Just to continue that, actually, it's a really, Dorit, thank you, really great point. How about combining other modalities to overcome the postmortem and the comorbidity problem, from variables to history, medical history, and implemented that into the analysis, so that it's in a way, if we know what happened through the life of the person at the end phase, maybe we can expect what was a healthy state. And I think Israel and Finland, North Europe and Israel are the leading, and you guys, are you trying to implement these things to analyze other information?
Definitely. The day before yesterday, I was sitting with a student that collected blood samples from a patient, and we got data about the patient record in order to make sure that we know how to exclude or include patients that are not, that will disturb with the analysis. And I think back to the question that Ido just addressed very nicely, sample size is critical. Many of the single cell paper don't have good sample size. And I think because of the heterogeneity, this is key issue. Do you agree, Ido?
Yeah, absolutely. We are, you know, we need to combine approaches which are, again, also remember that the speed and where we can collect a larger number increase, and the sequencing cost and data generation costs are going to drop two orders of magnitude even in the near future. So while we are collecting data that allows us to scale up, we need to understand in some approaches, we need to have more targeted approach, whether it's imaging or different FACS approaches, in order to have a statistical significant evaluation and deal with the complexity of the neurodegenerative diseases and obviously the challenges of the fact that patients are progressing in the disease for a long time, very difficult to stage them. There are many specific challenges with the neurodegeneration field, which other diseases just don't have. Autoimmunity, cancer, there is much more biomarkers, much more technology to do sampling from the site itself, and so we must be very imaginative. We must continually improve. And I think the exciting part is if you look at all other areas of the research, this is really in the very beginning of starting to understand the mechanisms and starting to understand the disease and the biomarkers. So in terms of progress, I think there's the most impactful progress to make, and I think we're starting there to move faster and faster.
Took more than 20 years to convince the community that the immune system has a role in neurodegenerative diseases. So I think that we are indeed at the beginning of this era now.
I also wanted to make the point that like in cancer, cohorts are very specifically defined. We can kind of break it up. Even within a specific cancer type, you can break it up pretty well. And here it seems like we can break up AD maybe into the progression stage, but there are all of these comorbidities, and there probably are finer stratifications. So I feel like maybe we don't understand yet the heterogeneity among patients, and maybe having a lot of patients can help start to understand and stratify cohorts potentially.
I think that many of the comorbidity factors in age-related Alzheimer are shared also by age-related cancer.
Yeah, I agree. That's one significant challenge. I'm going to have to drop off, so I want to thank everyone for the discussion, and you will continue with Michal, and really say, continue to build the next frontiers of HCA there in Vienna. I wish we could have been there with you, and Michal the same. I think we need to continue to communicate. So if people can share at the end the information, and I think many raise really important points, and I think we can really march on this together and make I think faster pace and really help each other progress to this very common and important good. So if there is a way to collect everyone that was on this break room, it would be wonderful to touch base on
Somebody here reminds me, in like 2 weeks, I will send out all of your emails to one another.
Ooh.
Just remind me.
I can remind you. I
Okay.
got it.
Katie, remind me.
Thank you. Bye, everyone.
Thanks, Tidor.
Bye.
So are there any still burning questions?
So getting back to Dorit's question about the differential expression, I just wanted to comment one thing. I think as you're starting, it sounds like you're starting your bioinformatics journey, but maybe I misunderstood that. I think a lot of these tools that are down there nowadays, like Soirot or ScanPy, have differential expression built in where you can do your you can get a first quick idea of what to do. So I think it's find marker genes in Soirot, or there's also options in ScanPy. But I think moving on to publication and getting further, you need to really think about what are the covariates that need to be included and all these things. And then your options will become more limited. And then pseudo bulk is still, I think, used quite a lot. And then the defining what you're gonna bulk together is quite an important step as well. Like which cells do you sort of group together and you're gonna compare to other things. But those are just my first thoughts. I'm relatively new. I'm coming from bulk as well, but we had a big discussion in our lab with all the bioinformaticians, like what are the best techniques? And this is sort of like start simple and move more complicated as is needed. And there's a lot of great tools out there that can really help you with it. But I don't know, maybe I misunderstood your question and your journey.
No, you did. I mean, we're starting to try out things like, so just to give more details, so my group is actually mostly interested in the process of brain maturation. So we're using these epileptic resections as sort of representing normal tissue, and we have samples of different ages, and we're trying to understand gene expression and how it changes over the course of that brain maturation in potentially mostly in pediatric patients in comparison to adults. And so yes, what you've just raised is like some of the problems we're coming into is a problem of sample size. Obviously, we don't have huge numbers of samples, but then also the problem of how to then maybe group the samples, say like into like how other groups have done it in the past, is group them into like epochs, like put samples of different similar ages together, and then to sort of try and get build more power and more numbers at each stage, and then do differential expression analysis using things like the DE-seq to pseudo bulking approaches, and to try and find like the patterns of genes that are showing similar patterns of change over time. So that's a sort of approach that we're trying at the moment. And obviously, as Michal raised, the more samples we get, the better. I'm just hoping that like with our few samples, I think we have about 10 libraries at the moment, whether that's spread across samples of different ages, whether we can at least start to tease out some differential expression in specific cell types. So we've chosen to pseudo bulk on the Allen Brain Atlas cell subtypes, so actually quite high resolution of different cell types, and then look at changes in gene expression across time. But it is challenging knowing whether we can trust the results with the low sample size.
Yeah.
I have to see that.
Another comment. I think too, there's great benefit in putting all of the data together, like forgetting the labels and doing some matrix decomposition. So methods like NMF or topic modeling. And I think that there are also pretty nice packages for this. And you'll get then just sort of like gene programs. You can see how they're expressed in your data. And, you know, just like looking at that expression on a UMAP, and then adding the labels, you know, sort of metadata, which patient it came from or which cell type it is, like you can put that information together. That can be really informative too. You can, and that can be really useful. Like for example, CD4 T cells don't have a lot of like very discrete cell states. Like differential expression wouldn't be very helpful there, but there are sometimes cell shifts that are more like along a trajectory and smooth. And this like topic modeling type of approach, it'll give you the same answer. Topic modeling, NMF, there's other factorization methods. You can learn a lot about gene. I know we talked about gene programs a lot here, but processes that are smoother transitions, which sometimes in immune cells are really important.
I think for macrophage, I think for the myeloid cells, you're absolutely, myeloid as compared to T cell, you're absolutely right. Because myeloid cells have a very wide spectrum of activity, whereas T cell are very limited.
Hi, two quick questions. The first is that, do you think cytokines released by immune cells are major contributors to the aggregation process? And the second is, by combining single cell technologies and imaging, do you think we would be able to gain insight into which proteins are in the aggregates? Because I don't think it is just a case of the usual suspects of alpha-synuclein, Huntington, amyloid, tau, etc.
I agree with you with both questions. For first, with respect to cytokine, there are not strong evidence to the direct effect of the cytokine on the A-beta accumulation. But generally, the state of inflammation of the brain worsening disease progression, accelerate disease progression. And there are enough evidence that the severity of the disease is correlated with the increase in local brain inflammation, which include TNF-alpha, IL-1 beta, and there is also now evidence for type 1 interferon, which exacerbate the disease. But there is no direct evidence that a specific cytokine increased the accumulation of this protein, of the proteonopathy. Secondly, with respect to the aggregate proteins themselves, in Alzheimer, we know that the size, there is a big difference between the monomer, the oligomer, and the plaques. And what is currently considered as the intermediate size, the oligomer, which are water soluble, are more toxic. And there are evidence that the recognition of this aggregate form is not by the sequence, but more by the structure, and they are recognized by what we call as disease-associated molecular pattern or DAMPs. So I think that we are, again, this is back to the question of the commonality. I think that what the cells are recognized as is very critical. I don't know, cannot tell you about specific alpha-synuclein or A-beta, but the structure of this misfolded protein is very critical.
Well, Michal, one question I have is immune system is a double-edged sword, and how are we going to handle is protective, and when we stimulated the hurtful effects, and we, at least I know very little to process of self-sustaining this healthy level of that, because it does a lot of things in the same time within the nervous system, and some of them beneficial, some of them harmful, and the balance is hard to control, I feel.
I think to call the immune system as a general concept is wrong, because the immune system is the body mechanism of maintenance and repair. Let's start with it. Now, in the context of the disease, it's very difficult to speak about the immune system. It depends what immune cells, where their location, what is their profile. So we cannot speak about general. What I think the emphasis is that if in the past we thought that the brain is self-contained organ, we now know that it's not self-contained and it's dependent on the dialogue with the immune system. So as long as you're healthy, the immune system is good, it's protecting you. When there is a pathological, I think the most important is to know that in neurodegenerative disease, unlike in autoimmune diseases, the immune system is not the immune system out of the brain is not a mirror image of what you have in the brain. And this explains why you have a local inflammation in Alzheimer, but treating with anti-inflammatory drug is not beneficial. So you're absolutely correct that it's double-edged sword, and our goal is to understand at what stage each of these cells is beneficial, at what is destructive, which cells we need into the brain, which cells we have to keep out of the brain. Total immune suppression is bad. So the therapy should, if we are harnessing the immune system, the therapy should be based on the understanding which immune cells are needed in the brain when you have a pathology, how to recruit them selectively, and how to make sure that they will acquire the right activity. Since we are short in time, I cannot give you a 2 hours lecture. How we understood now, better understood, we are not claiming that we understand everything, but how we understood the crosstalk between the brain and the immune system in health and disease.
Just in the Alzheimer disease, like the microglia depletion, every group who done that depends on the way it's done. We have different results, and that just suggests
because I think that I'm glad that you are raising it. I think that this is my personal view about the microglia. I think the microglia are resident immune cells. They are great for the brain as long as we are healthy. When you have a chronic disease, these cells may be early on, each individual cells may be early on responding in a beneficial way. But since you cannot resolve their activity, they can become detrimental. So if in a disease stage you are removing the microglia and you say that it's good for the brain, the only thing that it can tell you that the microglia at this stage are more destructive than beneficial. But to say that the microglia are not needed, it's a mistake. And I think one of you also raised the issue of the microglia about their activity. If you think that targeting the TREM2 is the target, although Ido and myself discovered that activation of microglia TREM2 dependent, I don't think that this is the target, but it may be wrong. This is my personal view. First, because it's not good in all models. Tauopathy and Alzheimer, they behave differently. Secondly, because maybe they are already senescent by the time. So I think that the microglia, targeting the microglia is more, we are not fully understand it. I don't want to give my own bias because this is not the stage now.
I think that's the whole models and what the experimental results, not conflicting, but the differences tell us it's a delicate system. And what you say makes so much sense to me as well.
I can give you a lot of information about it, but it's not a stage for me to say the microglia are good or bad. So I'm trying to avoid it. I have my own opinion. I'll be more than happy to discuss it, not in this frame. I think we need to close soon, so.
Yeah, so Michal, thank you. Christine here. Do you have a couple of minutes to stay on with the two scribes so we can finalize what we want to say in the report back?
Yeah, definitely.
Okay,
yes.
Great. Awesome. Well, everyone say thank you to Michal and Ido. Thank you, thank you. And Dorit, it was so good to see you and all of our virtual audience.
And you convinced me that it's summary in Vienna.
Very good. Well, thank you, everyone. We'll let our
Thank
scribes
you. It
and
was very
I'll
good.
get to work.
It was good discussion. Thank you.